We have Yuna Kim, who's going to give her talk tonight on the topic of, in the realm of ice and feathers, Antarctic summers with seabirds. So I'll just quickly introduce Yuna before we get started and hand over to her. Oh, and one thing, just a reminder, can you turn your phones off, please? So we're not distracted during Yuna's talk. Yeah, thank you. Ooh, lots of people checking their phones. Okay, so Dr. Yuna Kim is a seabird ecologist and the Seabird Project Coordinator at BirdLife Australia. She spent 3 summer seasons in Antarctica studying penguins and other seabirds, mainly focusing on their breeding ecology and foraging ecology. So tonight Yuna's going to tell us a bit about her adventures in Antarctica, and she's very much dressed appropriately for this talk tonight and for the current conditions. So yeah, I'd like to hand over to Yuna.
Thank you. Hello everyone, this is actually my 3rd time coming here speaking to you. I don't know exactly when I first came, but I did talk about Bruce Petrel in Cape Island and probably Broughton Island as well. And then second time I came here, I shared my experience in South Africa. I was working for BirdLife South Africa, working on with the fisheries, basically doing seabird bycatch mitigation work. And the last 3 years, I was lucky to go to Antarctica with the Korea Polar Research Institute. So today I'm going to share 3 years of my research. Rather than I show you research results, I wanted to show you more like how we did it, why we did it. So first of all, like Antarctica, it's such a big, part of the world. But I was on King George Island, which we need to fly from Sydney to Santiago in Chile. And then Chile is a very long country, so we had to fly from Santiago to Punta Arenas, which is the tip of, like the southern tip of Chile, for another 3.5 hours. And then from there, we chartered this little flight. I don't know, maybe 50 people can fit in. And the island airport is operated by Chile. So we fly there for another 3 hours. And then from there, so we are in the descent, sort of, airport is here, the Korean base is somewhere here. So we have to take a little boat in Zodiac to go across, depending on the weather. It can take sometimes 20 minutes, but it can take 45 minutes or 1 hour, depending on the wind direction too. So it is really far down under. We usually have about 3 days to get there, but once at Punta Arenas in Chile, I stayed for 5 days waiting because summertime, obviously the weather is cold but warm, and it's just, the airport runway is not really, like a modernized airport, it's just a pebble, so it can be very slippery to land, so I had to wait 5 days to get there. So the station I stayed is a research station operated by Korea Polar Research Institute. It's called King Sejong Station. It's one of the great king's name in Joseon Dynasty. And it's established in 1988, and it's very modernized, big station. They can accommodate 68 people maximum. And I was there as a summer crew for 3 years. It is really fun there. This is the main building at the back, and I'm wearing exactly the same costume I'm wearing tonight. So we have a dining hall. So this on the right hand side, that ground level is basically our dining hall. We have a greenhouse that we can grow fresh veggies, and we do Korean barbecue, obviously, time to time. And this is like a little supermarket. You don't need to pay anything. You can, if you miss your meal, you can go and get, something like instant noodle or cereals, whatever. It's really well managed, well sponsored research station. But it is a research station. We are not just having fun there. The downside, I would say, you need to share the room with other people. Usually female will share two. But as you can see, we have a desk, and we have actually separate office as well. And we have a lab and the common area where you can sit down and have a meeting. Or it's Antarctica, obviously, weather is not always very favorable to have outdoor activity. So if you are stuck in the station, then you can do a little seminar like this. We have a screen. But we also watch movie a lot of times too. But the research part. So basically, I was there for 3 years as an avian monitoring and research team leader of COPRI. So my other, Dr. Ji Kim and then Hyuk Jelly just finishing master's degree at one of the Korean university. We had very good, teammates and other assistants too. But basically, three of us working 2 years in a row. Collecting data. So our main research was monitoring nest distribution and breeding status of all avian species basically we can find in King George Island. But I should say it's actually a small part of King George Island, which is Baton Peninsula, because like I said, there's like Chilean base is there, and Uruguayan, and Polish, and also Turkish has a summer base in King George Island. So everybody has a sort of their own area to monitor. So our area is Antarctic Special Protection Area number 171. But so mainly we were working there. But my main research focus was the second part, the investigation of foraging behavior of avian top predators. So basically 4 species. Southern giant petrel, and then this is chinstrap. I will show you other species, gentoo penguin as well, and then the skua. And then the Dr. G. Kim was working on food chain using the DNA technology. So first, yes?
Just quickly, do you want to ask people not to take photos of the next couple of slides?
So some of the research parts, because we haven't published yet, and then it's not just my work. So if you don't take any photo, that will be good. And then I already said happy to record my talk, but few slides we will not put it on the YouTube and stuff. But basically this is very basic work we were doing, but I must say it's very important work we were doing for last 20 years. Like I was only there for 3 years, but Korea Polar Research Institute have been pinpoint, the nest basically. I left Korean names so you can guess all the birds. So this is a little bit controversial. First 2 species, because in Kopri they recognize 2 skua species. So the left one they say South Polar skua, and then the right one brown skua. Initially I went there and then I was thinking that this is same species, they look exactly the same. But then some people say, oh no, can you not see their head is a little bit like this, and they are a little bit bigger and more brown, and I don't know. And then I actually found the paper genetically they are actually not really distinctively, like other different species. But I will show you later the slide, they behave differently. So I don't know whether behavior, differences can qualify different species, but I will show you later. But this is a snow sheathbill. They are very funny birds, but they are the only one sort of doesn't have a webbed feet. And then southern giant petrel, and then this is Antarctic, the bottom, the second bottom, the red bill. That's Antarctic tern, and then black-bellied stone petrel, and then Wilson's stone petrel, and then Antarctic kelp crab. So a lot of people ask me, so how's all the penguins doing? Are they okay? Are they really decreasing? But our research shows actually gentoo penguin in ASPA 171 has been increased last few years, and then chinstrap penguins actually has been decreased. Both are actually listed as least concerns in IUCN Red List. But the trends are actually different. Like gentoo penguin, like our data shows, globally also they've been actually increased. But then chinstrap penguin actually has been decreased. But you can ask me why later. I wouldn't go too much details. But my focus was basically using tracking technology to identify where the penguins go and what they eat, and then how that's impacting their breeding performance. So I studied these 2 penguins, gentoo penguins and the chinstrap penguins. So using the tracking devices on the back, we could obviously get GPS points like latitudinal and longitudinal fixes, so can get foraging location, and with that we can, because it's logged by time, so we can get travel time, travel distances, and maximum distance they traveled. And then also some of the loggers we used can have the pressure sensor, so we can get the diving depth as well. So we were tracking from incubation period. Mostly it was at the end of the incubation period because they started their breeding, like laying eggs from middle of November, but mostly we were deployed in early December, so we were at the end of incubation period. But chick rearing period, we can get enough data. And then you can see penguins, some of them, we use the natural dye to number them because they look all same. And obviously, like we know, penguin can have tracking devices, then we know that's our study species, but study, the individuals, but some they lose because they actually dive really deep. So we do like water and the temperature of that, so the logo can fall out and we can still know which, birds we were handling before. And then also these birds we handled, we usually put like a PIT tags that you usually put into your pet, like a microchip, so we can continue monitoring in following years as well. Some people were concerned, like, how would you do that, like dying penguins like that, but it's actually more ethical because otherwise you will be, like, PIT tag is there, but you have to have like a little sticks to read at the back, like, and see, like, oh, is that you, like, keep going around and then they all get disturbed. So it's better to have like a dye the animals so you can identify easily, and then you only approach to the animals that you are handling. So we'll show you how we capture. We use this net only when they start running. So mostly they actually stay guarding their eggs and their chicks very well. So we can easily just grab them from like legs, or with usually chin strap penguins with the legs and the tail at the same time. But some animals we noticed once we handle them frequently, they recognize us and they started running away. Then we had to use those net to capture. And we also capture the chicks or eggs at the same time, because I will show you later the food chain. But they have so many predators, so once we just capture the adults and then leave the eggs or chicks there in the nest, other penguins will come and disturb, or skuas and shipsbill, they can all attack the eggs or chicks. So even if we are not doing any measurement, we will keep them in the, also obviously when the adults are guarding the chicks, it's to keep their temperature, so we actually have a little like a cooler bag to put them, safe from wind and predators. So this is a preliminary result. So gentoo penguin, chin strap penguin. So I didn't really show you the two penguins at the same time in the photo. Gentoo penguins are actually third biggest penguin after emperor penguin and king penguins. So they obviously have a heavier body mass than chin strap penguins. So they can actually dive deeper. And then also they are known to be the fastest swimmer. So we thought we will see a lot of differences, but then when we just get the average, time they traveled or distance they traveled, it's fairly similar between the species. And then also, I didn't really put the result here, but when we are looking at into the different breeding like a phase, because the incubation period, they meant to be incubating 3 to 5 days. And then chick rearing period, they meant to feed the chick every day, but we didn't really find the differences because, like I said, we were at the end of the incubation period, so birds are more alert and get ready to feed the chicks anytime soon. So I think that's why. But we saw more like individual variation, and that was also same across 3 years of data, rather than having like differences between species or different time. Or different breeding phase. It was more like individual variation was the biggest differences between this foraging behavior. And the southern giant petrel, this is my favorite species in Antarctica. Penguins are very, they are very cute, but they are very loud and very, I tried to find like nice words, but stinky. And then they shit had a lot of time, so it's like our clothes always get so dirty. And you probably noticed at the beginning when I show like our team members' photo, we are all wearing like a white PPE, like a protecting, like a suit for like avian influenza. And initially I was very against it because it's all plastic and one-off, and then you take all this, PP, and then we go every day nearly and throw away. So I was like, maybe we should not do that until we have outbreak. But then they said, no, we don't know how, these penguins can have symptoms or not. They can still have, highly pathogenic avian influenza, but they might be still fine, but we can maybe get something or transfer between. So they were advising us to wear. And the first time I won it, and then coming back to the station, I thought, this is actually great because I don't need to do the washing. Because I probably was at the beginning of the season, like you will probably wash it every 2 days, but then at the end of the season, you get used to the smell and then the mess. So you probably only wash it once a week or even like once in 2 weeks. And then you get told this by other colleagues who actually don't deal with the birds. But anyway, these flying birds are much cleaner and real birds, I should say. So we were also deploying the satellite tags on the back of their back using the Tessa tag, similar way we did for penguins. And then also you can notice that little thing on the leg as well. This is called geolocators. So that's more for long-term studies. They can log 3 years, just the logging actually light levels with the times. Then we will know the longitude and latitude fixes according to the sunrise and sunset time and day length. So, but yeah, these back on the back that satellite tags gives us the live location. So and it's very easy to catch. Like the birds, they are very loyal to their nest. So you can see we just get the eggs first because obviously we don't want to break it. So we put them in a cooler bag again to save and capture them. And then the chicks very well behave. But the non-breeders are actually a little bit difficult. So you can only see me there, but it was sort of 5 people basically slowly going. Because these birds are very big. They are similar to albatrosses, medium-sized albatrosses, 5 kilos, more than 5 kilos actually for males, and then female about 3.5 to under 5 kilos. But they need to sort of run to fly. So if you slowly get together, then you can capture them. So, I don't think it's working. It meant to be the video. It's not working. Anyway, so it was a little bit more difficult than capturing breeding southern giant petrel, but it was much easier. We don't need a net or something. And you actually don't want to use those nets for flying birds, their wings are very fragile. So, it will show as soon as we capture them, we make sure their wings are all folded nicely. So this is the sort of tracking, the non-breeders, male on the right and then female on the left. So King George Island at the beginning I showed you, it's at the western peninsula, was very close. So they actually go all the way down to western peninsula. But then the male was sort of just hang around in the King George Island. That was very interesting. And then the breeding pairs, you can see that red one is actually female. She was incubating for more than 2 weeks. And then during that time, male is going similar area where actually non-breeder went, and then foraging, and then they restore their body, and then they take turns. So we did track them, 16 of them. We deployed satellite tags and GPS tags, and then 20 on the lake with the geolocators. So we are analyzing it. We haven't actually seen where they actually go during non-breeding season, but it will be interesting because they actually have a distribution from Chile down to Antarctica. So it will be interesting. And then again, brown squaw and South Polar squaw, I really thought they could be the same species, but then I will show you how we capture them. They are very clever. So this little, so we use the legs. But we have actually our principal investigator is called Yuna Kim, and he actually can catch the flying bird with the hand, just the squaw. But it was amazing. But once you do this, they are very clever. They recognize obviously us, and second time we are catching is very difficult in the same way. So we will use this bow net. it meant to have some magnetic field, and then remotely, with using the remote control, we should be able to, close it. But in Antarctica, everything breaks. I don't know why. So we were having like this. So this is a little chick there, and obviously the parents is guarding. So we were slowly going close. So we really wanted to make sure we are close enough, so when it's actually closing, we don't harm their wings. So obviously for flying birds, like hurting wings is the end of their life. So we were very cautious, but most of the time we were just not able to catch. But first time we were using those, I don't know what to call, a special tool. Like it's very easy, but then they obviously learn very clever birds. I liked them actually. Now I look back and then it was fun. But then once you go there working for 3 hours and then you don't catch any birds and coming back home, it's just like, I need a beer. So brown squam, as you can see, can you say these birds, seabirds, they didn't even touch the sea. So it was very interesting because this is where they have this pink hot area. That's where actually the penguins are. So they can easily get just the penguin eggs and chicks. So they don't even just touch the sea water. But then this is a South Polar squirrel. They actually don't spend much time at penguin colony, and then they forage in the sea. So after looking at this tracking data, I was thinking, they might be really different species, but it could be like people say brown squirrels are actually bigger than the South Polar squirrel, and then maybe bigger, individuals can occupy or more territorial, around the penguin colony. So maybe that's why they show these differences. So we need to look into it more. But something we really need to understand in relation to their movement, and movement is their diet. So we use DNA metabarcoding. This is basically at the supermarket, you have those little barcodes, right? Similar way, if you have any animals or any basically living creatures with DNA, they did any DNA work, they can upload into gene bank, and then you do use a certain marker, and then you can actually match to see which species could be close to what you are seeing in the genetic bands. So we collected post samples basically from any Antarctic bird species to do the DNA metabarcoding, and the result will be actually presented at the AOC, the Australasian Ornithological Conference in November in Perth. But we sort of know what they are eating by observation. So this is a gentoo penguin feeding. I don't think it's working. Gentoo penguin feeding the chick. So you can actually see they say penguin milk. Oh. No? Oh, it's
click, I think.
It's disappeared from here.
It's very red, so you can see they heavily rely on krills, because the krill color basically they are feeding, and sometimes they spill it, and you can actually see even eyes of krills and then some fish and things. If it's not working, we can pass.
Nope.
Yeah, that's okay. We can, but maybe next slide also. Oh, it doesn't even show. Next slide meant to be, I think, Seabird. Snowy Seabird.
Because there's nothing on the next slide.
Oh no, it all goes blank. Maybe this is stopped working. Yeah, so I was filming a snow Seabird was eating penguin chicks, and it was very painful to watch because basically they were eating belly of a little penguin chick, and then chick was still alive, and he was keep looking at the birds, and then, oh my God, I wish I can save, the little chick, but obviously we shouldn't interrupt like that. Yeah, and we also observed the southern giant petrel was eating gentle penguin chicks as well. So we. Oh, it's too fast. You can hear the little chick begging for food. And then you can see like a fish also scales and things you can see. And then this is actually squirrel eating the, maybe you can play. I believe that was the seal. We don't know which seal, but squirrel is actually getting all the blame for Antarctic birds and avian influenza outbreak because of their scavenging behavior. And we've seen them eating all sorts of things. Even they eat, the other nest squirrels, eggs and things. They will eat anything, everything. And this is the video, very heartbreaking. But their views is very sharp, and when they are actually taking the eggs, they like a fork, they will poke it and then fly away, and then they will eat it. It happens every day, basically. But they also raise their kids during the time. Same for squirrels. So in the happy feed, you see like squirrel was like showing as a bad guys, but then they also need to survive to eat. So basically, before I joined the team, they were doing mostly just the penguin research, and they are still getting, the attention, basically, all the funding basically are put for penguin monitoring and penguin tracking work. But I managed to get more funding to track southern giant petrel and squirrels, because we really need to understand other birds at the same time they are breeding. And then something I want to include is the seals as well. We have Antarctic fur seal, this is crab eater, that's leopard seal, and then that's elephant seal, southern elephant seal. And so we want to understand the whole ecosystem using penguins. So obviously with the DNA metabarcoding, we will have all other fish and other marine probably animals we could see in their diet. But something we should be aware of is DNA metabarcoding is still new, and unless especially fish species, unless those fish are commercially very valuable, no genetic informations are available. I think Antarctic probably fish is even understudied, so it could be still limited, but this DNA metabarcoding will basically open up broaden our understanding of whole food chain in Antarctica. So that's really about it. I must say thanks to our principal investigator, Dr. Jung Kim and Dr. Ji Kim. There are so many Dr. Kims in and we had so many fieldwork assistants, and also these wintering crews are basically maintaining all stations and help us with our research, and really had a great time there. So I am now no longer work for Korea Polar Research Institute, but working for BirdLife Australia. So I thought I will take a little bit of opportunity to advertise our upcoming event. So as you probably know, 19th of June is World Albatross Day. So we are having webinar. So you can use this QR code to register for the webinar. It will be for 1 hour from 7 to 8, but I will be presenting about albatross conservation in general for 20 minutes. And then Tanya is the avian influenza response coordinator who actually developed this AviFlu map. So you can see where the outbreaks are, I should say not just outbreak, it is reported the influenza cases with Deakin University and other Wildlife Australia. So she will be also talk about some of the albatross disease, because this year's Albatross Day theme is the effects of disease. And many of you, I believe, are a member of BirdLife Australia, and then probably get this text appeal in your mail or email. And so I'm very happy to see Guldspettre. Probably a lot of you supported Guldspettre project last 20 years or even more. So this year's theme for text appeal is marine birds. So if you want to reduce your text, you can also use QR code to donate, or you can actually ring or email BirdLife if you are particularly, interested in supporting your local seabirds. Then you can email them saying, I want to particularly donate for Guldspettre project too. That's it, I think. Yeah.